Assalamualaikum and hi everyone. Let's continue with subtopic 9.3 Fertilization and Fetal Development. At the end of this lesson, students should be able to define embryogenesis, state developmental stages from zygote to the formation of morula, blastocyst, and gastrula through cleavage. Define organogenesis and state the organ formed from each germ layers during organogenesis. Embryogenesis is a process by which human embryo forms and develops. The zygote undergoes a series of cell division and cell differentiation to form embryo and occur during the early stages of development. Once fertilized, the ovum is referred to as a zygote, a single diploid cell. The zygote undergoes mitotic divisions with no significant growth, a process known as cleavage, forming cells called blastomeres. The first division produces 2 daughter cells, then 4 cells, and 8 cells. A morula is formed when the cells consist of 16 or more cells forming a solid sphere of ball of cells. The morula has the same size as the original zygote and still surrounded by zona pellucida. As the morula grows into more than 100 cells, a fluid-filled cavity forms in the center, transforming it into a ball of cells, known as blastula or blastocyst. Blastocyst refers as a fluid-filled hollow sphere ball of cells. When the blastocyst cells migrate inwards, it leads to the formation of layers of cells called germ layers. The embryo now is known as gastrula. This diagram summarizes the stages of embryogenesis. After ovulation, fertilization occurs and a zygote is formed. The zygote then undergoes cleavage, forming 2 cell stage, 4 cell stage, 8 cell stage, and eventually forming a morula. Morula undergoes more cleavage, forming more than 100 cells. It transforms into blastocyst, a fluid-filled hollow balls of cells. Blastocyst then implanted into the wall of endometrium. Blastocyst undergoes process gastrulation, forming 3 germ layers. Now it is called gastrula. This picture shows you the cleavage process from 2 cell stage to 8 cell stage. When the cells reach 16 cells or more, it is called a morula. Morula has the same size as the zygote but with more cells. The morula develops into blastocyst or blastula. The blastocyst consists of outer layer called trophoblast, a cavity called blastosome, and a cluster of cells called inner cell mass. The blastocyst will be implanted into the endometrium wall between day 6 to day 9 after fertilization. This picture shows the blastocyst is embedded into the endometrium wall. This picture shows the blastocyst is embedded into the endometrium wall. As implantation occurs, gastrulation proceeds. At the inner cell mass of the blastocyst, the cells move inward and form 3 layers of cells which are ectoderm, mesoderm, and endoderm. Next stage after gastrulation is organogenesis. Organogenesis is the formation of organs from the 3 germ layers, which are ectoderm, endoderm, and mesoderm. The layer ectoderm is the outermost primary germ layer in an animal embryo. It develops into the nervous system, epidermis, and sweat glands. Mesoderm is the middle primary germ layer in animal embryo. It develops into the reproductive system, kidneys, muscle, bones, skin, blood, and blood vessels. It develops into the reproductive system, kidneys, muscle, bones, skin, blood, and blood vessels. Endoderm is the innermost primary germ layers in an animal embryo. It develops into the lungs, liver, the lining of the digestive organ, and some endocrine glands. This slide summarizes the organogenesis formed from the three germ layers. The ectoderm will form the skin cells of epidermis, neuron of brain, pigment cell. The mesoderm will form the cardiac muscle, skeletal muscle cells, tubule cells of the kidney, red blood cells, and smooth muscle. The endoderm will form the lung cell, thyroid cell, and pancreatic cell. That's all for this chapter. Thank you.